Hi, Bradley.
Hello,
Hi.
how are you?
Fine.
Good.
I have some couple of updates and some idea to introduce a new model, I think so, for cell lineage analysis this week.
All right.
Maybe this could be next year GSOC project, or we could work internally after the GSOC.
Yeah.
And I'm writing. I think I've shared two mails to you right now about docs of DevoNet and DevoLearn separate docs. I've started the draft manuscript actually
Okay.
on DevoLearn, DevoNet, SAM individually.
All right. Did you send them to in the chat here?
Yeah.
I don't see them yet. I kind of joined late, or after you did, so.
Yeah.
All right, there we go. Okay, so this is the doc. This is DevoLearn. This is the abstract. And then, okay, this is the DevoLearn paper.
Another one is also coming right up. There are three papers actually. One was DevoNet, which the model which I have proposed in my proposal, SAM, and DevoLearn. Because DevoLearn we don't have any paper or preprint there. Preprint I think so. So I thought of making one. At least we could host our research as a preprint, so we can continue on it. So that's the reason I started writing them. Actually, I extracted the weights of the weights file .pth for SAM model and DevoNet also. I have compared it with the unit model and got some accuracies around 88% and 92%. I have mentioned in the SAM paper.
Okay.
And another thing was Bradley. Just a minute. I'm sharing my screen. Could you see it?
Yes.
Right now, we don't have any. I came across this video actually 2 days back.
Okay.
This thing got me some new idea. I'm going to keep that in chat. Right now, we don't have any models for directly plotting the cell lineage analysis. But
Right.
in this video, now in this video they are directly segmenting they are making custom data set. Let me show it to you. Yeah, if you can see this image, they are making custom data set and fine tuning on the Detectron 2 model from Meta AI to get cells, what are alpha granules, etc. In C. elegans case, we can do this thing. We can make our own data set about 200 or 300 images, and if we fine tune on that model, we can get the cell lineage analysis. Like if a picture is there, right now, this picture is there directly. It can mention this belongs to A cell or E cell according to lineage analysis directly, like this in this video it is there, you can see here, right?
Yeah.
Alpha granule, these are mentioned. Actually it was fine tuned on this tutorial can be useful, and we can use that in C. elegans for cell lineage analysis. Right now we have a model on cell lineage analysis, but it doesn't tell which cell belongs to what. Which cell named as A cell, E cell. It just tells the count, like these many A cells are there, E cells are there, D cells are there. If you want to create our own data set, naming all these conventions. Actually, in this video, they have mentioned how to make their our own data set and how to fine tune it also.
Okay.
Actually, couple of days before GSOC, I don't remember, you asked or Gordon asked me, mentioned me about right now. There is no segmentation model which represents a cell and tells like it is a cell on top of it. I was thinking like
Right.
this. If we can fine tune this, it could be a first model. We can show the cell segmentation and the naming also in the same time.
Yeah.
Right now, this week I have completely worked on the manuscript. I have over you here. Any comments would be useful by next meeting. I think so. I will add some more matter and I will add the pictures also. Right now, I was struggling with the pictures, like plotting all this stuff. I
Yeah.
will try to add those pictures. And before, like I have seen some of people are working on the SAM fine tuning on cell images. So I was thinking to host it as a preprint. At least we could create something and show we are working on this thing, because so many people are working on this thing, SAM fine tuning,
Yeah.
and our work and their work are approximately same. I think so. I mean, uh, but if we could host a preprint, that would be useful. At least like we can claim this thing we are working on it right now as GSOC project, etc.
Yeah. Yeah, that would be fine. Uh,
Uh,
yeah, so let's look at the third paper you sent. So you sent the DevoLearn, the microsam, and then the third one is the
DevoNet.
DevoNet. Yeah. So this is this is DevoNet. What's the difference between the DevoNet and the microsam? The microsam, is this the general case or?
MicroSAM is like a fine-tuning SAM model,
Yeah.
uh, which was given by Meta AI. But DevoNet is like completely different. It is like whatever the proposal I have wrote, right? It's completely dependent on that. I was not only working on one model right now. I was parallelly working on two models. I was trying to work it out in the same manner. In one model, it can segment perfectly at zero-shot performance. Zero-shot performance means whatever image if you give it, it will obviously segments it, and it can developmental criteria of a cell like volume, 3D volume, area, and timestamp, their position, etc.
Yeah.
But we are lacking in the cell image analysis. I think so. Like extracting which cell is named as A cell, B cell, etc. I really don't know the nomenclature of it, how we name those cells. But if we can crack this with this data, I mean, I have kept the video also. Feel free to check it out when you are free, Bradley. I think this could be next year GSOC project, or I think we have participated in Hacktoberfest. I think so.
Yeah, we do.
Like we can host this year.
Okay.
As a project, this one, maybe people can work on this could be useful. I mean, if you see this if you see that video, you'll get an understanding like what I was mentioning. But we need to fine tune it on our own data set. We need to make our own data set of this thing, bounding boxes, naming those cells. That's the reason I was thinking this for next GSOC project next year. I could help you with this and these models also.
Yeah, I think that would be great. We should definitely shoot for a Hacktoberfest for to get people working on it. So yeah, we have the data for the cell types too. We have like the cell nomenclatures. It's just in a tabular data form. So I'll send that to you.
You need a video data as well, and a tabular data mentioning like the position and the cell name. That would be useful.
Yeah, I think actually we do have some videos. I don't know how far it goes through the cell lineage tree, but we do have some videos. I don't know. I can't remember where they are. I'll have to find them. But I know we have the tabular data though for the cell lineage, like the nomenclature and everything. So I can send that to you. We can put together something. I don't know what we want to do with them. I mean, in terms of like hosting them or extracting information out of them.
Yeah.
But yeah, I
If you
think
just
that's a good idea.
if you just fine tune this on Detectron 2 model from Meta AI, sorry, I will share the model also. I will share the
All right.
Detectron. This is the link actually. This is the link I have shared here. Detectron 2 from Meta AI. This could be useful for object segmentation and naming it also. If we fine tune this on C. elegans data on lineage analysis, it could mention it like this. Like here, if you open that repository, you can see that. Just a minute, I will share my screen.
Okay.
see, right now in this image, it was segmenting it, and it was also mentioning that it is sky, it is skill, etc.
Yeah.
If we are able to fine tune this model on C. elegans data, it could mention like what is the cell name according to the cell position to XYZ. We can get the cell image analysis and the position of the cell also. We can fine tune and we can predict like DevoLearn's model only. Have checked though everywhere, like this model wasn't there, and this model was trained on lots of data, so it could give a zero shot performance on C. elegans data also. If we have data of around 700 images or near to 1000 images of C. elegans at different number of cells, it could give better results as model. I thought of working on, but I have a lot in my plate right now, writing these 3 manuscripts, making a clear documentation. So maybe after completing this project, I will contribute to it, or I will start with you another project in the meantime after this project.
Okay, that sounds good. Yeah, this looks.
Or we can post it as Hacktoberfest or next year GSOC project also. Maybe some other students may take part in it.
Yeah, I think definitely Hacktoberfest would be good.
Okay, thank you.
So let's shoot for that, and then I can get the data to you, and we can set up something for people to use. Like, we'll have it all set up on GitHub, and then basically Hacktoberfest works by people going to the GitHub repository, contributing, and then we give them some prize for participating a certain amount, and then that's pretty much it. But then they submit work and get credit. So yeah, I think that'd be good. I think 2 years ago we did Hacktoberfest. It was actually pretty good. So yeah.
I think it's my time you did this Hacktoberfest. I think so.
Yeah.
So not our Mayuk or Mainak, I really don't remember. I have participated in it.
Yeah.
But anyways, really, okay, those are my updates. I have completely worked on the manuscript of these 3 papers, comparing the models, getting the results. Maybe by next week, once you read these papers, what I am writing, I have shared links with you personally and in the chat also if they are there. If you can guide me through this, I will complete these papers within 2 weeks and we could end this project. I will clear the documentation work also.
Okay.
Maybe we can work on other new models if we need.
Okay, that sounds good. Yeah. So for the figures, it's just like the graphs of data, or what are you trying to do?
I'm making the graphs and the segmentation images, and actually there is a library called WNB. I was integrating our model with WNB, which creates the experiment. WNB, which tracks the experiment. Okay, see this WNB site?
Yeah.
But we can try track our experiment, like how the learning rate is increasing, how the loss function is working, accuracy, etc. So that would be useful there.
Yeah. Yeah. Well, let's try to get a couple of key graphs put into those drafts. I'll look them over this week, and I'll give you some feedback, and then hopefully we can get them together soon. Or yeah.
Right now I haven't added. I just added the abstract introduction and methodology I was working on. So I will update all paper maybe by this week, and I will give you at least a manuscript. I think you remember Jyoti Swarup. He started. He worked with this. Maybe he will help you with making a clear journal paper. Actually, he was expert in that. He will help you with that. I couldn't join the meeting, I think so, but yeah, he will help you with that.
All right, sounds good. Yeah, thank you.
Okay. Those are my updates. Thank you.
Okay, thank you. Yeah, so thank you, Sushmanth, for the updates. Let's see, we had a couple people joining us, Dick and Lukas, and Jesse was here when I came in, and Himanshu's here, and we'll get to Himanshu's update now. It looks like Susan had a meeting with her advisor this morning, so she won't be here. Himanshu had a couple of things that he mentioned. He mentioned that custom annotations would be required for the tabular data. Is that in reference to the the stuff that I said that was in the tables? I guess, well, Sushmanth was talking about a certain type of model that can incorporate those data or be specific to those data. So segment with respect to the locations of those specific cells. So is that a custom would you consider it to be a custom annotation, Sushmanth? Or?
Yeah, custom annotations are needed actually. The data right now we have just the microscopic images, but if we have according to the cell position, if we have the name of the cell also, it would be useful.
Yeah.
We can try that. Bradley, please check out the video I have shared here. There is a YouTube link between this. Maybe, yeah.
Yeah,
please, this would be useful. You can understand the whole thing.
Okay.
It explained how to how to fine tune the detectron model, how to make our own data using the open source library. Those all things are mentioned. So maybe, yeah.
All right, that sounds good. And then Himanshu said there is a C underscore raw underscore data CSV I'm working on, which was used in DevoGraph 2. It has the data set of the lineage tree and it has coordinates for timestamp and size. So yeah, I think that was one of the files I was thinking of. So that's on the GitHub repository, in the GitHub repository for DevoWorm, not DevoLearn. And
that's actually just a CSV file. We don't have the video image data actually. That's the problem.
Well, I have some separate data. It's not on our GitHub repository. In our GitHub repository, I can get that together. I have it somewhere, but there's like a video where it actually has, and then not really. I can't remember the quality. I don't think it's like the highest quality video because it's pretty old, but it has like the identities of the cells marked in the image. I don't know if that'll help, but I can provide that as well. All right. So thank you. Himanshu, would you like to give your update?
Yeah. Am I audible?
Yes.
Is it visible?
Yes.
Okay. So right now I'm working with the persistent homology part of the DOGL graph neural net. So I was figuring out what kind of persistent diagrams we could get and what kind of topological analysis would be good on the data set that we have currently. So the brief overview of that was to get node attributes and create a node map, take all the views of the graph and apply some kind of filtration function which will give us the persistent diagram. And after that, it's just aggregation which helps us to incorporate it into a graph neural net. So the main part over here is what kind of filtration functions which we use, and that gives us the corresponding persistent diagram. So last week I had given a last week I had given a tutorial on this low star image filtration function, which was used on the data set which was for image segmentation, and it gave pretty good results for the lifetime and of the cell, and after smoothing the data, it gave a diagram similar to this one, which was seen over here. And also after we plotted the points, this was the output, which gave us low image, the critical points in the image. So I was working on the data set, the raw data set, which was which had the timestamp for the cells and that coordinates and also the size. And I was working on the I plotted the entire thing, and I was just working on the filtration function, which would be helpful for this kind of data set. And right now, I've googled a lot of things, but I couldn't find the example which was exactly for a use case. So I was just figuring 2-3 things out. One of which was dense and sparse filtration. but I've got an error over this. I'm still working on it. I would continue doing this, and after that, I'll figure out a way to incorporate this TOGL repository, which was like the main paper which we are referring to for the graph neural net, and like look into this repository and find out how to use its components which are important, and we can refactor and use it, especially like the persistent homology part, like this persistent analysis, and another thing like this analyze persistent homology statistics part. Also, like I couldn't work much last week because I have an eye infection, and it's kind of like a sty, but it's much worse, and it's in the inner eyelid, so I can't like look into screens that much, which was really hard.
Oh yeah.
So that's my update. I'll be working on this, and after that I'll figure out a way to do the TDA part.
Okay, yeah, that sounds good. It looks like you're getting there. So yeah, let's keep going. That's good. I guess by the end, I guess we should have at least sort of an analysis, just kind of sort of not like a full formal analysis, but just a few a few analyses to show kind of what the graphs should look like, what the output should look like. So if they're tables, that sort of thing. Because I don't know if we know, like, or I don't know necessarily what should be reported from an analysis like this. Like, what are the takeaways from it, and how can we make it useful?
Yeah.
Yeah, so that's good. I imagine there's like a subset of graphs that you come up with at the end, and that's your answer, as it were. So yeah, that'd be interesting to see. Okay, so that's good. Do you have anything else to report, or?
No, this is an update. I'll work on it and get back to you next week.
All right, sounds good. Yeah. Thank you, Himanshu.
Thanks.
All right, so that was Himanshu's update. Looks like he's making progress on the. This is like the topological data analysis stuff that he's been working on, and this is basically the C. elegans embryo. We also had a training set with larval images. Have you been using that as well, Himanshu?
Yeah, like the larva images, I'd used the different, like the data set which we use for membrane segmentation. This was the one.
Yeah.
Like the cell. So that is what I use for the filtration function for the image, but for the graph for the graph data set I'm working on.
Okay.
Yeah.
All right.
So for the graph data set, there will be a similar filtration function, but it would be dependent on the distances corresponding to it. So like these points are similar together, and for example, this point is much farther, and so there will be like a distance function which will give like it will create a matrix with respect to its neighboring points, and like figure out a way to such that for each timestamp and for each point or like data in the diagram of each cell, basically like the AB cell, the ABA cell, and the ABAL cell, etc. how they are related to each other with respect to its topology.
Yeah. That sounds good. Okay, yeah, look forward to see you keep working on it. Looks good. Yeah, so thank you, Himanshu. All right, so that's our GSOC updates. Now we turn to Dick and Lukas and myself. We're working on this paper. So we have the paper, and Dick's been reading it over. I don't know if he had some comments or what he wanted to do there. Okay, what does that mean?
The ultimate, the one before the last.
Okay. So what do you need from, is it from me or from Lukas? What do you need?
I need you two to agree on a version to send to me and Natalie, and we'll go over it tonight.
Oh, okay. Well, yeah, I think, yeah, I incorporated the changes that Lukas made, and I'll send you what I have tonight. I've corrected all the references as well in it, so good. And I'll send that tonight.
Okay, good.
All right. Any other comments about that? I don't think they're... Lukas, do you have anything?
Yeah, why don't you send it to Susan too for her comment?
Oh, right, yeah. Oh, Jesse asked which paper. This is a paper we've been working on. Last several weeks have been talking about it on differentiation codes. So
Yeah, it's we're attempting to map the differentiation code onto the genome.
right. All right, that sounds good, and I'll get you that stuff today.
Okay, great.
Yeah. All right. So actually, I can go over it a little bit in the meeting. Let me share my screen. Okay. So this is the paper, abstract introduction. Basically, it goes through this idea of differentiation codes, which is where
Can you double the size there?
Yeah. So this is figure 1, which is restylized from the original diagram. So it's basically where you have, in this case, we're talking about a binary code that is based off of the lineage tree or of differentiation events in the embryo. So you have these.
Still see
you know.
Bradley, there's still a problem. Look at the caption.
Okay.
The caption mentions that it's colored.
Oh, right. I got to take that out.
Either take it out or put the colors in.
Yeah, yeah. And so the idea is that you have the lineage tree basically reorganized so that you have things that get smaller or contraction waves going to the left and things that get larger expansion waves going to the right. And so that's basically this whole idea of a differentiation tree or a differentiation code. And so in animals that are not C. elegans, like the regulatory embryos, regulative embryos of, say, like axolotl or mouse, these circles are tissues. Whereas in C. elegans, and we did a paper on this back in 2016, these circles are cells of different size. And so we can actually reorganize the lineage tree in this way and then put a code attached to the different constituents. So there's a 0, a 1, a 0, 0, a 0, 1, a 1, 0, and a 1, 1. And then from that, we can do different, that provides a code throughout the embryo. And then but that's only based on cells and tissues. That's not based on the genome. So that's a different thing that we're trying to investigate in this paper. And we do some things with gene regulatory networks. We're looking at how genes are expressed and the sorts of things that they produce when they're expressed to further produce proteins, which should show up in amounts, basically the proteins that we might expect to be involved in development. So we go through some of these gene regulatory networks and their products here. We talk about gene duplication events and other things that can add to this network, add to its complexity. And then we basically link that back to some of the differentiation codes. So there's this binary code that you get from the cells, and you can link that back to these genetic regulatory networks. Then we kind of move into an analysis. So there's some theoretical background. We move into an analysis of epigenomic mechanisms and proteomic mechanisms. The epigenomic mechanisms are these presence of these CG motifs within candidate genes, and then the proteins are a protein list that was provided in Dick and Natalie's book on what we should expect to find in terms of proteins important in differentiation. So we have those proteins, we have those counts for 4 species. We have, I don't know if I have this table here. It's towards the back. Okay. So this was actually put together by Lukas. And this is so what we did was Lukas went into GenBank and he did a BLAST-P, which is a protein search for different proteins associated with a certain species and a certain identity. So we have this list of proteins. We have 4 species that we're looking for, and we get a count of proteins or matches that return. And those are basically proteins that have an identity. I think there's, you actually did a count. I can't remember. I have the methodology written in here, but basically we had to get a count for each protein. And, there's certain things you have to do to make sure that you don't get false positives and things like that. But basically, we get a difference in the count across different species and across different types of development. So in this table here, we have our proteins. They range from, through a number of pathways. There are things like the JUNC pathway, the MAP and MAPK pathway, PLC, PKC, Rho and ROC, and WNT. And so these represent different pathways in development. And so we should see, depending on the type of development, differences in these counts. And then, of course, we have C. cerevisiae, which is yeast. And yeast doesn't exhibit any sort of development at all. It's a single-cell organism that lives in colonies. They're metazoans, which means they're part of the animal lineage, but they're not, they don't have a development. So we have yeast, we have mouse, Mus musculus, which is this regulative development. We have Drosophila melanogaster and C. elegans, which are different forms of mosaic development. And so their development is different in certain ways, but they have the same type of mosaic development that we've talked about with respect to the differentiation code and differentiation tree. So you can see that there's some similarities and differences in the protein counts. It's kind of hard. There isn't a clear signal, but you do get cases where the same type of development shares some protein counts that are very high with respect to the other 2 examples. You get cases like CDC42 where everything has a lot of that protein, but you get cases where, indeed, like DVL, Drosophila melanogaster has 2, and then the other taxa have 0. And this is in terms of the count that we're making. And then ECAD has 14 in C. elegans, nothing in the rest. So you actually do get species-specific protein expression here in ECAD. And there may be another case where that's the, oh yeah, RAC is also like that. And WINT for that matter. Of course, there are some, we actually did check for some of the, homologs for different species. So like, for example, the WINT pathway is important in Drosophila melanogaster, but it's not the same protein as WINT in C. elegans. So we didn't count it as being there. But you can see that mouse and C. cerevisiae or yeast have a count of 0. So these are different, these are going to be a little bit, when you make a count in bioinformatics, sometimes you have to make certain assumptions. And we list the assumptions in the paper. But a lot of times, you get these kinds of comparisons you couldn't do otherwise. So I think
I have a question.
Yeah.
Are we allowed to include a snap of the figure 8.13 from Richard's book so that they could see what's the 23 proteins in the figure?
Yes, we could ask for permission. We'll probably get it.
Okay. Yeah, we could add it. Yeah. I mean, we cite it, so it's kind of people can go to the book. Like we cite the book, but also the figure. So that's usually enough, but we could put the figure in as well. If there's confusion in the reviewers have some confusion about it, we might do it. Then these are the protein normalized counts. So these are graphs that kind of compare. This one, A, is where we compare C. elegans and Drosophila melanogaster with, with yeast in the top. So you can see there's some that are yeast, and this is just like compared to, if C. elegans and Drosophila, which are in blue and red, are in the positive territory, and yeast are in the negative territory, which is gray, then that means that these things are expressed in both our mosaic development examples, but not our no development example. Sometimes that happens, and sometimes you get cases where Drosophila resembles yeast, or C. elegans resembles yeast, for different proteins. Then you have this case here, which is not yeast, but for mouse. So again, you have the C. elegans example in blue, the Drosophila example in red, and then the mouse example in gray. And so you can see that the same holds true. A lot of times you get Drosophila and mouse sharing proteins or having a similar trend in the count, and then C. elegans being different. And so, that's again a case of sort of an intersection of, what we can capture with these kind of counts, different proteins being used for different things, and which may be that it's not really the effective development type isn't really the same, or could be just species specific things like C. elegans is a very specific mechanism for something, or Drosophila is a very specific mechanism for something. So that's the protein counts. We also did a bunch of candidate genes for the proteins. So in C. elegans, you can go to there are different resources that you can use for this. There's Wormbase, there's NCBI, then there's a literature. And so you can go to all those sources and find analogs to different proteins. So there are a number of papers where, for example, you could look up a protein like MAPK, and you can get candidate genes for that protein. So in C. elegans, that's actually quite common, and there's a pretty decent set of papers where you can go through and they've identified the genes for the protein. That's not true of every species, but in C. elegans we can do this. And so having done that for, what's that?
Yeah, when you have a table that's split between pages,
Yeah.
you can do one of two things. Either put a page break in, or there's a command for duplicating the header row for the table on each page.
Okay.
Makes it more readable.
Right. Okay. So this is basically what we got. So what happened here was we have these gene we can go back when we know what the candidate genes are, get gene sequences from NCBI, and then analyze those gene sequences for the CG motifs. And we get a count of CG motifs across these different genes, but across also the protein categories. And then we have what I determine information count, which is basically looking at the variation of different patterns within that count. So we can take short windows of 12 bases and see how many CG repeats we have within those little windows of DNA. And the reason to do that is because these CG repeats tend to occur in islands. And those islands are sort of tell you something about how it's regulating itself or how the gene is regulated and the places where you can have greater regulation of the gene. So this is based on the literature. It's a little bit different mechanism than what happens in mammals, but you still get this sort of effect. So this is a summary of those window counts, and these are the raw counts for these. So you get these motifs in different genes, and there's actually, if you look across different genes for different proteins, so for example, RME6 and RAB5, both involved in CK1, they have different counts. And they have different, well, not quite the different, they have about the same information, but they have different counts. And some of that is an artifact of different lengths of the sequence. But this information count is probably the most informative thing in that analysis. And so we can use that as well to tell us something about how the gene is regulated and how that relates to the protein. And so there's further analysis of how that, how those things relate to one another in different proteins. Then we build these gene regulatory networks, which are constructs that, where you sort the genes, and sometimes they're on different chromosomes, sometimes they're on the same chromosome, and you sort them by their information content. The things that have a higher information content are on the right, and the things with lower information content are on the left. And the ones on the right are sort of attenuating the things on the left. And then you have a protein count here. And so this organization should give you a higher or lower protein count. And so I give a couple examples here for different genes or different proteins with their constituent genes in this network. And then there's a discussion of that. And then that maps to the differentiation tree by being sort of the thing that's in this cell, the thing inside the cell that's controlling potentially this state. So if you go to A, one of these networks is in cell A, and then the cell A divides into 0, 0, and 0, 1. So it serves as sort of a switch between these two states. And so the question is how do we determine the threshold for switching between the two cells? And that's that's kind of not a very good way to say that, but there's a lot to consider here. And so there's a little bit more here on some of the configurations of these gene regulatory networks with respect to the differentiation tree and what you might see inside of, for each gene as it evolves. This is a phylogeny where you have different configurations of the gene regulatory network. So this tells us that there's an evolution over, there's change over evolution, but there's also change in development for these different networks, and they translate into this tree and then ultimately to that code that I showed you. And so that's pretty much the paper. I know that was a kind of a whirlwind tour, but I'll send out a copy today and we'll look it over and see. And the deadline is Thursday, so yeah, we'll have some time. We might ask for an extension, perhaps, or I don't think there will be that much to change, maybe. So we'll see. Any comments or questions about that? All right. So yeah, that's a lot of work there. Thanks to Lukas for doing such a good job on the protein counts and helping us put this together. He's very, and there's a lot of work that sort of that he produced in terms of like what kinds of things are, like he came up with these searches and he found a lot of things that were related to other things. So that's some of that is included in the paper and some of that is not. So there's a lot more data that was generated from this analysis that might be useful for other things, and I'm not sure what it's useful for necessarily. I mean, it's a lot of good work. I don't know. There might be some other opportunity there to look at some of these things with fresh eyes.
We might have to ask the OpenWorm Foundation. I could talk to one of the guys from there. There might be some useful data specifically because they need, they make Jupyter notebooks and like models and stuff like that. I can't do models with that, but I think it's useful for the community. I have to ask the person in OpenWorm Foundation or maybe a elegans lab. I only know this lab, so not too much.
Yeah. Well, we can do a lot of that stuff. We can put together like Jupyter notebooks and things, and we have done that in the past where we've archived stuff like that. So I mean, we could do that too, and we could do it through, we kind of have an arm's length relationship to the OpenWorm Foundation in terms of that stuff because they do a lot of stuff with, largely with like neurophysiology. I mean, that's a lot of their data sets involved. But our data sets, we have a lot of development specific stuff. So we might be able to put together like a public data set where we kind of synthesize a lot of the stuff you were finding. Because, there's a lot of interest in comparative analyses. And so some of the stuff you brought up, I think, was very useful to comparative analysis. And that might be something worth looking at more. But I'll look through it later. Right now, I want to get this paper submitted and through the system.
Oh, yeah, of course.
Yeah.
All
right. Yeah, Jesse. Yeah, you can take a look at it. So yeah, well, okay. So that's good. So now I want to turn to some other. Let's see. So we talked about differentiation code paper. I have this interesting paper here. Let me share my screen. It's a classic paper. It's on, we'll see in a minute. So this is a classic paper on Drosophila mosaics, and this is from 1972. So this is going to have a lot of hand-drawn pictures in it. Because that's what people used to do in biology. They used to draw pictures of hand-drawn things, and some of it was really good. It's kind of a lost art, but so there are no like images in here, microscopy images. It's just drawings, apparently. This is actually about talking about. So the headline here is by making genetic mosaics. So genetic mosaics are basically a term for different types of mutant genotypes. By making genetic mosaics and constructing embryonic fate maps, it is possible to locate the anatomical site of abnormalities affecting behavior. So In C. elegans, you have this as well, where you can create different mutants. You can target different genes, and you can like mutate those genes, and then you can backcross the worms so that they have this gene always, this mutant gene always expressed. And then sometimes those mutations have an effect on behavior. So if you have something that is like, a certain gene you want to know the function of, you can create a defined mutant for that mutation in that gene, and it can have all sorts of strange behavioral effects. It can be like a slow crawler. It can be like it can crawl backwards. These sorts of things. And they're very, you see this even, you can do this in mouse even in some cases. But this is mainly restricted to organisms where gene is really tightly related to behavior. So it's like C. elegans and Drosophila as well. So you have this is their paper where they kind of introduced this idea. So this is mapping of behavior in Drosophila mosaics. Let me zoom into the paper a little bit. So they talk about the circuit components of behavior, from sensory receptors to central nervous system to effector muscles, are constructed under direction of the genes. So we have now, in the last couple of years, we've gotten a number of different subconnectomes of Drosophila. We've gotten a couple of the different circuits for behavior and for motor behavior and sensory behavior. So we're kind of figuring out what the connectome map looks like. Now the role of genes in that is a little less clear, but we know at least what that connection map looks like. A mutation affecting the structure or function of any of these components may alter behavior. So you can get these defined mutants that sort of, they can change the behavior of this connectome, whether it's by knocking out some neurons or altering the way that the connections are made or whatever. Using classical genetic recombination mapping, the locus of the mutant gene with the one-dimensional chromosomal array can be determined. So they did this genetic recombination mapping technique, and they're able to get sort of localize the location of this mutant. This is before they had widespread gene sequencing, by the way. So this is a technique they had to use to get at identifying these mutants. They couldn't just sequence the gene and look at the mutation. It was much harder to find out about what was going on in the gene at the time. It is another problem to identify the focus of the genetic alteration. That is the anatomical site at which the mutant gene exerts its primary effect. So this is where you have this, how does the gene act? It's always been a question of, where does the gene act? What does it do? What's the function? So like in the paper that I just showed you, we have candidate genes for specific proteins, and even that's not a tissue, that's a protein. But some of those genes are, associated with other proteins as well. So it's not a clean-cut association. But nevertheless, that's the data that we have. So we have to kind of go with that and see. So that's what we deal with. A lot of genes are not specific to one protein, or there's not a one gene, one protein mapping. And so that gets even worse with different tissues and behaviors. So now we have a bunch of interactions working under the hood to, that's the great mystery of genes to phenotype or genes to behavior. And so they talk about this mosaic technique that they use. Mosaics are composite individuals in which some tissues are mutant and the rest have a normal genotype, making it possible to identify which part must be mutant to produce the altered behavior. So this is an example of the Drosophila mosaics. So what they do is they take, I guess they could do this in development, but they basically take different cells and put them in, I guess, I don't know how they make these, but, well, I guess you can do it through the germline, putting in mutations where they're only expressed in certain cells. But in this case, what we have these adult Drosophila, and they have this mosaic phenotype. So one part of this is expressing is derived from one cell line, and the other part is expressed from another cell line. So in this case, body parts derived from the XX cell line, which is shaded, are female, and then parts derived from the XO, or unshaded, are male, and also express recessive mutant characters uncovered by the loss of the other X. So basically they have two different genotypes in them. The cells on this side of the organism have one genotype. The cells on this side of the organism have another genotype. And so that means that you're going to have the mutant expressed sort of locally. So you can have it like down the midline, but you can also have it in different organs. You can have it in the head and in the wings. You can have it in the wings and maybe parts of the body, etc., etc. So you can find, you can refine this technique to express one genotype in one part of the body. I think this is maybe a lost art as well, because now we have things like CRISPR, or we have other types of retroviral constructs that you can just put into certain tissues and you can actually get expression of genes locally. But this is something that is sort of a tradition in genetics, creating these mosaic flies. And so that means that they can actually create a map of the body parts. So this is a really fascinating diagram here. This is where you have the head, the thorax, and the abdomen, and you have these body parts that are like taken apart. It's almost like if you ever built a model car, and you have like the parts laid out on the table before you build it, and they're just kind of sitting there all in pieces, and you have to identify them before you can start. Because you have to say, how many wheels do I have, how many doors, and so forth. And so this is what it looks like, kind of. You have these parts that are labeled, but they're all kind of splayed out, the legs and the forearms and the head and everything. It's just kind of out like this. And so these are the external body parts. Each part is formed independently from an imaginal disc present in the larva. So during metamorphosis, the parts develop into the adult form and are sutured together to form the exoskeleton. So this is just the outer shell of the organism, but it's exploded like this to show the parts. And so then they are able to make a map of surface landmarks from this diagram. And they're basically trying to analyze these ipsilaterally and contralaterally across the body. And they're trying to find some sort of distance between these landmarks. So you do this in the normal phenotype, and you do this in the mutant phenotype. And now we have these mosaics. So the normal phenotype and the mutant phenotype are in the same organism. It's just that when you go to a certain part of the organism, it's normal or it's a mutant. And so you're measuring these landmarks across these different parts. So the idea is that if you're in a mutant part of the phenotype and you measure that distance between the two landmarks, it should be different than, say, like if you were to do this under normal conditions. If there's a difference between the normal and the mutant for the same set of locations, then you can say that that mutation has an effect on the phenotype. It's kind of a hard thing to wrap your head around, but basically you're looking at the effect of the mutation on the phenotype, on the shape of the body. Since you have it in the same individual, you can look within the individual, you can look at the other side of the organism, if that's where the normal versus mutant part is, and you can compare those two. You shouldn't see a lot of difference because there isn't a lot of asymmetry, fluctuating asymmetry. But also, if you look across organisms to a normal versus a mutant. So there are different ways you can measure this. So this is the formation of a mosaic fly by loss of one X chromosome. So this is where this happens in the egg, where you knock out an X chromosome in one cell, but you keep it in the other cell. And then as the cells proliferate, you get this set of knockouts, and you get this set of wild-type cells, which don't have the knockout. This is where the cells proliferate, and then they go on to form this outer ring, and so this persists in the outer ring where you get this whole section of the organism with the X knocked out, and then this whole other section where everything is intact. And then that leads to this embryo where you have a whole region where this is going to be the one side of the embryo where you have the mosaic genotype, one side not having the X, the other side having the X. And so that's how that works in development. You start with one cell. You can start with one cell and modify it, and that can persist throughout development. And that's what it looks like at the end. So it's very interesting. And then they build this fate map. So this is what a fate map looks like in Drosophila blastoderm. So you have this, this is constructed by mosaic mapping. This just shows kind of the body parts here and how the fate unfolds in the context of the anatomy and of the parts of the different parts of the embryo. And so this just shows that fate map and how they build that from the embryo. And so they, so then, okay, so this is a principle of mosaic mapping, just showing how you have to, find the distance between these different landmarks and what they should look like in the mutant versus the wild type cells. And so you can actually, so mosaic mapping, you have to generate a series of lines across the phenotype. And, they're going to be different with the different effects on the phenotype. And then this is so they look at a number of different mutants here. So they look at the visual receptor mutants, hyperkinetic mutants, and then this mutation called drop dead. Drosophila is really unique in that it has a lot of really quirky names for its mutations or its genes, and some of these mutants have similar names. So this is called drop dead, and this is, I guess, a mutation that is involved with the worm dropping dead, I guess. The drop dead mutant flies initially develop and behave normally, but at some time an individual begins to walk in an uncoordinated manner, and within a few hours dies. So this is a syndrome that they express over, at some point, there's an onset and it goes pretty quickly. After an initial lag, the number of survivors drops exponentially with a half-life of about 2 days. So this is what this looks like. So this is something that we can perhaps look at markers on the phenotype to see if we can identify this mutant before we actually observe this drop-dead behavior. And so they're looking at different places on the phenotype where you might be able to identify the mutant. So if you have a drop-dead mutant, you should be able to identify it from the actual phenotype. And so it turns out that there's a lot of variation, and it's kind of hard to do. But yeah. And then they talked about mapping interacting foci. So again, there are a lot of interactions that are underlying this. So you have to consider mapping these interactions between genes, which is kind of, a challenge even today with a lot of the data that we have, which is much better than this data. There are different, so the various configurations of the mosaic boundary, we have to know kind of where the boundary is so we can do this mapping properly. And genetic interactions make that even harder. So that's the point here. So this is, then this is another mutant, wings up. So you can see that there are a lot of mutants in Drosophila as well as C. elegans, where you have these very clear effects. And they're single point mutations, so it makes it even easier to identify them. The problem is identifying them in the phenotype. You can identify the behavior, but are there other markers in the phenotype? And it's kind of hard to get at. And so these are some interesting pictures of the orientations of the boundaries of these mosaics. So you have differences in these boundaries. They're not very clear in the diagrams that they were showing. It's just like a line down the midline where there's specific organs that have one genotype versus another. In reality, and especially in the in the embryo you have, it's kind of more of a contour map, I guess, and it's not as clear cut. So, you have a lot of, that boundary isn't always as clear as you would like, so it's harder to do this analysis. So this is a really interesting paper. It's from 1972 and involves a lot of drawings. So, yeah. So, I don't know if you have any questions, comments? Okay, so that's all I have for today. I'll get that paper out to people this evening. All right. Thanks for attending and have a good week. Okay, now I'd like to present on a paper that is related to the differentiation code discussion we had earlier in the meeting. So this paper is on gene regulatory networks and C. elegans, or more specifically feed-forward regulatory logic. And so the genetic regulatory networks we introduce in the paper are largely about gene-gene interactions and the genes that go into producing these proteins, and specifically the protein states based on these counts. In this case, though, they're considering the temporal dimensions of a gene regulatory network, something we're not really explicitly getting into. But it turns out there are a lot of ways you can build gene regulatory networks, and one of them is to build a network of genes to see, what regulates what. But another thing you can do is do some sort of dynamical simulation of the activity. And so that's what this paper concerns more. So let's go over this a little bit in diagrammatic form. So a gene regulatory network is basically a statement of regulation. So if we think about like a statement of regulation, it's a graphical statement of regulation. And that's basically what we're trying to do. There are a lot of ways this can be done. I'll put graphical in here. A lot of ways this can be done, and in the literature there are a number of examples. For one, you can have genes, such as they are, in a relationship. So you can say, first of all, you can just have plain gene networks, and these are not regulatory things at all, but they're just kind of like genes that are related to one another. So this might have an output, this might have an output, but this is like measuring, say, gene-gene interactions. And so you can make a statement that gene A, gene B, and gene C are all related to one another. They might be associated with a certain protein, they might be associated with a certain phenotype. Gene A is related to gene B, so it has an effect on gene B. Gene B is related to gene C, and gene C is related to gene A. And it may be that they interact in some way so that they have some additive or multiplicative effect. It may be that you notice in your data that if gene A is highly regulated, then gene B is less. There's less of gene B around, at least the product of gene B. We're always looking at gene products. But we can also look at the sequence and make statements as well. So we might hypothesize then that the regulation of gene A leads to the downregulation of gene B, or the upregulation of gene A leads to the downregulation of gene B. But you can make this statement about gene-gene interactions, and it's just basically a static snapshot. So this is a static snapshot of a gene regulatory network. But of course, that's not all genes do. They regulate, they produce products. They don't just sit there with information in them, or with, a lot of people don't like the information metaphor, but I think it's apt because basically in this context, it's telling other components of the network what to do. So if we have A, B, and C, they're regulated. So that means that something is making copies of this gene in terms of transcripts, and those transcripts are going off and doing things. They may be making a protein, they might be regulating other genes. So in this case, A has a promoter, which is A, B has a promoter, which is B. C has a promoter, which is a C. And we can actually add more. These are called regulatory elements. Actually, I need to go to the next page. So we have A, B, and C. We have A as the regulator, B is the regulator, C is the regulator. And these are all promoters. And we can add other types of regulatory elements. But for now, let's just put a promoter in there. And we can say that A regulates B. But A regulates B through the activity of this promoter, turning it on, producing more of A. And then in B, we're going to add another element, which is B prime. We'll call that a repressor. And so then this promoter is turned on somehow. Then it turns on A, makes copies of itself. And by virtue of making copies of itself, it engages with the repressor of B. And then that represses the activity of B. So B makes less copies of itself, or fewer copies of itself. Then that affects C, of course, because C cannot be promoted. So there's no input to C. And then that can actually have an effect on A, because that can actually maybe turn off the input to the promoter, or it might be able to activate a repression element, this repressor, in some way, and turn down A. Or, paradoxically, it could turn up A. If C is, for example, was repressing A, and there's less of C, and maybe there's an environmental stimulus that's promoting A, then the environmental stimulus wins and you get a lot of A, but less B and C. And then there's an output to this, to making a protein or something. But these regulatory networks can occur in different ways. It can exist in different ways. And so this is kind of what they're getting at in this paper. In this paper, though, they're talking also about feed-forward regulatory logic. So they're talking about a feed-forward system, which is where you're going from A to B, or from B to C. There's no feedback. C to A would be feedback. A to B and B to C would be feed-forward. And this regulatory logic, which means that there's a certain logic where, certain elements have a certain effect, and you can actually model this as a set of logic gates or a set of logical statements, controls the specification of differentiation transition and terminal cell fate during C. elegans endoderm development. So they're looking at the endodermal cells in C. elegans, and as we talked about, the C. elegans lineage tree is deterministic in that it always produces the same types of cells. So the cells hold, they have this sort of terminal fate where they produce a certain cell type. But that cell type is primed early on, and it's always going to, that sublineage is always going to become a certain type of cell. So there's the specification of differentiation transition. The cell is specified transcriptionally, and then it differentiates as the phenotype. So the abstract reads, the architecture of gene regulatory networks determines the specificity and fidelity of developmental outcomes. We report that the core regulatory circuitry for endoderm development in C. elegans operates through a transcriptional cascade consisting of 6 sequentially expressed GATA-type factors that act in a recursive set of interlocked feed-forward modules. So these are 6 GATA-type factors. These are just genes. They're sequentially expressed. They act in what they call a recursive series of interlocked feed-forward modules. So that means that the modules are nested or something like that. They're self-referential, and that in that sense they're interlocked. So their behavior, it's kind of like this ABC motif that I showed you here. There's a feedback element here. But depending on, I can organize this so that it's just simply nested in a way that's functionally relevant. So there are different ways you can organize the topology of these networks, different ways you can turn them on and turn them off, and then you have a logic. So this A, B, and C would be like gene names. They would have, each gene would have some sort of role in that network, and we could model, basically, this type of phenomenon. This structure results in sequential redundancy, in which removal of a single factor or multiple alternative factors in the cascade leads to a mild or no effect on gut development. So they're looking at gut development specifically. Whereas elimination of any two sequential factors invariably causes a strong phenotype. So this is interesting because if you go back to our network here, this is only 3 elements. But let's say we had something like a 8-element network, and I'm going to turn this into a larger sort of diagram here. Let's suppose I had 8 factors here, 4, and I'm just getting different inputs from different places here. This is an input, this is an input. All right, so this is 8 factors. We have, let's see, I'm going to do this just to be kind of a feed-forward kind of guy. So this is our A, this is our B. Our B goes to C. It also goes to this one here. We haven't named it yet. A and C are getting inputs from the environment. D is here, E is here. Yeah, we can do it that way. F is here, G is here, and H is here. So you can see that B affects both C and H, or C and G. Then C affects both D and E. E and C both affect D. D has an effect on F. This I just put this B in here. This is from the environment. So let's get rid of that. And then F affects G as well as B, and then G affects H. And so now this is our 8-factor network. Now what we're going to do is start knocking out factors, and we're going to see what the output looks like. So you can have this sort of redundancy where you have something where you can have an alternate path through the network. So say I knocked out F. If I knocked out F, I could still go from A to B to G to H and then do an output. So it would disable part of this network, but it would still work. If I knocked out E, that might even have less of an effect on the whole network because I can go from A to B to C to D to F, which is not knocked out in this case, to G to H, or from A to B to G to H. And C, if E is knocked out, it has really no effect. But let's suppose that I have E and F both knocked out. Well, then I recover the effect of F and the effect of E, which is minimal. And so there may be some modulatory effect in D that E is having, but it's mitigated by the knocking out of F. So that has sort of the same effect as F. But let's knock out F and G. In that case, this circuit really becomes non-functional because A and B can go to G and H, but if G is knocked out, then it can't go anywhere. Similarly, this whole part of the circuit, C, D, E, and F, don't have an output, don't have an outlet. So basically, it's this part of the network kind of operating on its own, not finding a way to express itself, and then H being the thing that doesn't have anything that's regulating it, and the whole network falls apart. So this is how you can get these differential effects. You have to find the right location to make these mutations. But the way this specific network is set up, one mutation knocking out one factor doesn't have much of an effect, but knocking out 2 factors can. And so this is where I was trying to get at here, which is that these cascades, which are these sequential activation of factors, can have, depending on the topology of the network, can have wildly different effects. So this structure results in sequential redundancy, which I showed you because you have sequences that if you can knock out something and find an alternate path. The phenotypic strength is successfully predicted with a computational model based on the timing and levels of transcriptional states. So if we go back to our first diagram, we see that there's this promoter, repressor, it's producing an output of this gene, so the products of this gene have an output. The promoter tends to make more copies. The repressor brings the copy number down. And so this is what they're getting at here, is that you can actually, the timing and levels of transcriptional states can serve to, because some genes are dependent on genes that are upstream, they can actually have these kinds of effects. So you can have a repressive effect without actually having like a repressor regulatory element. You can just, it's just that the product isn't there to regulate it. So they are actually looking at this in terms of timing and levels of transcriptional states, previous levels of transcriptional states. We found that one factor in the middle of the cascade, N1, which straddles the distinct events of specification and differentiation, functions in both processes. Finally, we reveal roles for key GATA factors in establishing spatial regulatory state domains by repressing other fates, thereby defining boundaries in the digestive tract. So this is where this gene regulatory network acts to establish sort of these state domains, which are, where you have different areas that are basically upregulated or downregulated. And this is a different state than what would be, say, inside the gut. So if you're at the boundary of the gut, your gene regulatory network would have a different state than inside the gut. So you can define boundaries in this way. Our findings provide a paradigm that could account for the genetic redundancy observed in many developmental regulatory systems. So they kind of go through this. They talk about the common genetic toolkit for the generation of diverse animal forms. GATA transcription factors play a conserved role in the development of diverse cell types, including those of the endoderm, the first of the three germ layers to have evolved during the late Precambrian era. In the dipoblastic phyllonidarians and poriferans, GATA factors are specifically expressed in the endoderm or in endoderm-related cells, suggesting that these transcription regulators may have driven the development of the endoderm germ layer and gastrulation at the dawn of metazoan evolution. These GATA pathways are really kind of important for regulating cell state. And so there's an interaction with some of the proteins that we were talking about in the differentiation code paper with some of these other networks. And so we don't consider those networks specifically, but we're sort of proposing a very general mechanism for this. So as it turns out, there are a number of mechanisms that people are identifying in C. elegans, for example, for specific cells that lead to the differentiation of different tissues or cell types in C. elegans. So as I said, different parts of C. elegans, the different tissues, have a very deep origin in the lineage tree. So the endoderm arises from a single blastomere formed at the 8-cell stage, known as the E cell. So there's a sublineage called the E sublineage. And the E sublineage is actually going to go on to form a lot of what they call the endoderm, which is the gut and some other parts of the intestine and that sort of area of the animal. Endoderm development is driven by 3 pairs of duplicated genes encoding GATA-like transcription factors. And they talk about MED1 and MED2, END13 and ELT27. And so these are canonical factors, and these are divergent factors. So this is where they kind of identify the different factors involved in this this gene regulatory network. So they talk about sequential redundancy, and this suggests feed-forward regulatory circuitry. So this is kind of what I was showing before. I showed kind of a loose example of it's not based on this paper, but it's based on like this idea of having different feed-forward configurations of the factors so that there is this redundancy. The redundancy that you see in those kind of networks is evident of the phenotypes of single mutants of most of the genes expressed in the C. elegans endoderm GRN, which show either no overt phenotype or weakly penetrant phenotype, meaning that the effect of that mutant isn't great. And so, you don't see it in, like, penetrance means what proportion of the population are expressing this factor. And then, if it's not apparent in the phenotype, then it doesn't necessarily have a strong effect. So this is where they kind of talk about some of these things. You can remove genes and you can see what their effect is. Like we saw in the paper with mosaicism in Drosophila. Okay, so all of these findings were consistent with the three-tiered model of redundant transcription factor pairs. Temporally resolved transcriptomic analyses revealed that the GATA factor encoding genes are sequentially activated in the mesendoderm regulatory pathway. This observation is consistent with the alternate possibility that rather than the pairs of factors acting together at specific tiers in the cascade, each factor is redundant with its immediate upstream or downstream factor in a continuously sequential cascade, or what they call sequential redundancy. So this is an example here, what they give of these different tiers. So they have A plus B, C plus D, E plus F, and this is MED12, N13, and ELT27. These are the analogs in the C. elegans genome, as based on their theoretical sort of formulation of this. So you see what they're doing is they're taking what I did, which was A through F. They're just kind of making up like boxes, and they're saying that they have this additive effect at each tier. And then they kind of go through this with mapping genes to those boxes. So they say MED1 and 2 are A plus B, C plus D is N1 and N3, and then E and F is ELT2 and ELT7. Furthermore, you have this feed-forward network A, B, and C. A can also affect C. A can affect B. B can affect C. And then you have this mapping to the genes. So MED2 has an effect on MED1. MED1 has an effect on N3. MED2 has an effect on N3. MED1 has an effect on N1. So it's really kind of like you have this linear pathway from MED2 to MED1, N3 to N1, ELT7 to ELT2, but you also have these shortcuts that are feed forward from END1 to ELT2, for example, or from MED2 to N3. And so that's the topology of their regulatory network. And they show the viability and some of these other factors that they're measuring in these different mutants. So these are defined mutants in this graph here in C. So they have these different effects of genes. So they have the single genes, single mutants, MED2, MED1, N3. This is just where you have that mutant in the your mutant phenotype. Then you have these sequential, so you have mutants in double mutants, basically, where you have mutations in two genes, but they're in the same regulatory network. So they're sequential. So you have MED2 and MED1. So there's a sequential effect there, and you have a double mutant. And that shows that it has a different viability than the others. Then you have alternate. So you have MED2 and N1. So this is where they're not directly affecting one another. They're sort of indirectly affecting one another. But you have double mutants in this case, where you have two of these genes that are mutated, but they're not directly affecting one another. And this just shows the viability here. So you can see that there's differences in viability. You have these terminal differentiation genes down here, and they're affected by this pathway. So you have two different topologies. You have in J, you have this feed-forward pathway with the feedback at ELT27, and then this one in K where you have more feedbacks and more shortcuts across the feed-forward components of the network. And you see that there's a fraction of viable animals is higher, while they're both pretty high. But there's this other factor, early expression of ELT2 relative to wild type. This is the predicted amount. And so you can see that in these organisms, there's much less early expression of ELT2, and in this case, it's more diffuse in the sequential case. So this is an interesting set of graphs. I don't know if it really differentiates the two topologies that much, but that's basically the relationship we would expect to see on the terminally differentiated genes. So then they develop a computational model. They carry out an in silico model of these GRNs, which means they model it in a computer. They perturb the two models in different ways so they can simulate these mutations. They're computing the expression levels of different genes, and the computed ELT2 expression levels in the tiered model only weakly correlated with the phenotypes of the mutant combinations. So this tiered model is the one where you have these tiers, whereas I guess this, which one is the tiered model here? The J is the tiered model, where you have the tiers that kind of move down one by one, and then this is more of the feed forward. So this is actually the sequential feed forward model. So the tiered one is J, and the sequential is K. So we'll model both of these in a computer. So in contrast, when the feed-forward model was tested, our corrupted results predicted that ELT2 expression would occur with delayed onset in all single mutants and in mutants lacking alternate pairs of GATA factors. However, ELT2 expression was predicted to be completely abrogated in double mutants in which sequential members of the endoderm cascade are removed. So this is in the sequential network. If you remove those, there's an effect. And this is consistent with their pronounced developmental defects. So you can do this where you can take expected results from phenotypes that you observe. You can model different types of network topologies, different types of relationships, and you could work out what you should expect from those different topologies, and then you can make sort of an educated guess as to what the actual genetic regulatory network looks like. Then there's synergistic requirements and cross-regulatory interactions of END1, ELT7, and ELT2. So, you have different effects of different genes. Sometimes they act synergistically, which means they both need to be there, all three need to be there to have the effect, but it has a greater effect than just the additive effect that we might normally expect. And so they work together and they have an effect on phenotype. As described above, both ELT7 and ELT2 single mutants contain a fully differentiated gut, which shows a contiguous lumen from the pharynx to the rectum, which is surrounded by cells of normal differentiating morphology. So this means that these mutations did not have an effect independently. However, ELT7 and ELT2 double mutants, which means that they're now in a position to act synergistically since they're both in the same genotype. These double mutants invariably lacked both a defined gut lumen and at least some overtly differentiated gut cells, i.e. an apparent sporadic all or none to block gut differentiation along the length of the animal. Although differentiation was highly defective in the absence of ELT2 and ELT7, patches of well-differentiated gut were nonetheless evident. Moreover, many terminal differentiation genes continue to express in the absence of ELT2 and ELT7. So this means that eliminating the functions of these genes had little effect on the expression of, say, ACT5, which is another gene involved in this, a gene encoding an actin isoform required for microvilli formation. So these observations suggest that at least one additional factor, in addition to the ELTs, may activate gut differentiation. So this is an important point because it's not only that these genes have to act synergistically to have an effect, or they both need to be mutants and to have an effect, but we don't know the full extent of this network. It's an incomplete network, and so there are a lot of things that are coming from the outside that are regulating it. This is beyond like maybe the environmental effects on the network. So there's transduction of signals from the environment of the cell, or you have other chemical signals that activate this. You can also have other genes that are involved. And so this is something that, is a matter of inference. It's not just that, these genes are self-contained and that's that. These are just candidate genes. And so we have to work with what we have in terms of the candidates. Sometimes they're predictive and sometimes they're less predictive. Taken together, our results suggest that N1 and possibly N3 is poised at the interface between specification and differentiation in the cascade, placing it at the crux of this major developmental transition. N1, acting with N3, regulates specification of the E lineage, whereas N1, acting with ELT7 and ELT2, control differentiation of the intestine. And so ELT2 and ELT7 collaborate to safeguard intestinal cell fate. They basically they sort of enforce that fate in the cells, no matter what else is going on. So this is graphs on the synergistic actions, this ELT2 antagonizing N1 expression, and kind of gives this idea of what these different genes in this regulatory network do. So there are a lot of things it does in establishing the boundaries of the system, the different cells of the system, different components. And this is, of course, another set of examples where you're knocking out different things, and the actions may be of outside factors, like PAL1 and Wnt, MAPK, SRC. So you can see that there are different things that this gene network just doesn't operate in isolation. It's affecting other things. It's being affected by other developmental factors, and it's affecting other developmental factors. So it can shut down other developmental factors. It can be turned on by, say, like Wnt cascades or MAP signaling or whatever, and this can have an effect on the output of this gene regulatory network. So that's all I had to talk about with that paper. Hopefully that's food for thought. And thank you for listening.